Okay, shall we continue, guys? You have a question?
No.
What happened?
No, I'm just talking to.
Oh, you're talking across the room. You're sending signals. All right, let's continue on, guys. All right, so we have seen how the genome is organized. Again, you have your 3' end without the poly A, you have your 5', and these are the various proteins, and your enzymes, the different proteases, that's where they'll cut. So let's have an example and see how yellow fever virus works, okay? So for yellow fever virus, we have positive single-stranded RNA genome. It makes 10 different virus proteins, 3 of them are structural, and 7 are non-structural, okay? Now, these 3 of these non-structural proteins, they have enzymatic activity, okay? And the 3 final structure proteins are included in the mature virion are all precursor proteins, okay? So what that means is they need to be processed to make the mature virion, okay? So the ANC and PRM, precursor M protein. So again, here's the genome, different proteins, and you'll see some of them are forming the capsid. There's the PRM protein, envelope protein. This protein has dual functions, so protease and a helicase, and a methyltransferase polymerase, okay? So these are the different proteins that are going to be synthesized. And these are the various proteases that are viral proteases as well as cellular proteases that are going to cut at these specific sites. So you have your NS2B, NS3, a signalase enzyme from ER, furin is present in the ER lumen, and this is unknown. Basically, they don't know which enzyme cuts at that spot, okay? Now, if you guys want to see what these different proteins do, this is the detail of the proteins. So you have your structural protein and your non-structural proteins. What I want you to focus on is the proteins that I've underlined, okay? So for example, anchor protein. So this is the anchored capsid protein. This is the precursor to the capsid protein, okay? Once the sequence gets inserted into the membrane, it's cleaved by the viral proteinase. Then you have your C protein, which basically helps with encapsulation of the genomic RNA. Your PRM is the precursor protein. It binds to the E-glycoprotein, so envelope protein. It inhibits the membrane from fusing with the Golgi membranes as it goes through Golgi. And once it gets to the right site, it gets cleaved to form the M protein, okay? And that's going to release this PR peptide from the M protein. Your E protein is your major envelope glycoprotein. M protein is the small envelope glycoprotein, okay? We don't know what the function is for this protein, but your E protein, just like we have seen for other viruses, it binds to the receptors and allows for the fusion of the viral envelope with the cell membrane, okay? Then you have your various non-structural proteins, so they're just labeled NS. So NS1 all the way to NS5 with a couple of variants thrown in, okay? I want you to focus on NS2B, which is the proteinase that cleaves the viral polyprotein. You have your NS3, again, which is a proteinase that's involved in polyprotein cleavage, okay? This also is a component of the RNA replicase complex, okay? As well as it has nucleoside triphosphate as well as helicase activities. So this helps basically with multiple things for that virus, okay? Lastly, NS5 is your RNA-dependent RNA polymerase. It also has the methyl transferase activity. So what that means is it can actually add methyl groups to the 5' end of the genome. So 5' cap structure, okay? Now let's have a look and see how these proteins are organized, and then we'll continue from there. First thing is, your E protein will direct how this virion binds to the receptors and how it fuses with the membrane. It's a type 1 membrane protein, and normally it's found as a dimer, okay? And that's important, and I'll explain that in a minute. Now, this protein will lay parallel to the lipid bilayer of the envelope instead of protruding, so as a result, you have a much smoother surface. You have two major domains here, or three major domains, domain 1, 2, and 3. The domain 2 is used for formation of a dimer. So here's your 3 domains, and here is your other part of the protein. Okay, that's domain 3 for the second subunit. Okay, now see what's happening here. Here's your fusion peptide. Okay, your domain 2 will form a dimer interface. It has that fusion peptide, and this formation of the dimer actually protects it from the aqueous environment to prevent premature fusion of that envelope, okay? Domain 3 here is used for binding of the receptors, so it has an immunoglobulin-like folds, and domain 1 basically joins this is domain 3, domain 1 joins your domain 3 and domain 2, okay? So again, your fusion peptide is here, and then you have whole another protein here, and as a result, this dimer prevents premature insertion or activation of that fusion peptide, okay? So the question then becomes, how does it attach and enter the cells? We actually haven't really identified a specific cellular receptor for these viruses, okay? But once the attachment does happen, the entry is endocytosis mediated with clathrin-coated vesicles. So once you start to endocytose, you're going to form these clathrin structures on that membrane invagination. Once that's inside, the clathrin coat comes off, and now you have a membrane structure that contains your virus. But there might be an alternative entry mechanism where if the virus is bound to an antibody, it can enter the cells that express the Fc receptors on the antibody. So immunoglobulin Fc receptor. So if this was an antibody structure, which one is the Fc domain? This part right here? Yeah. So that's the Fc structure on the antibodies, and it has specific receptors on some type of cells which recognize that structure. So what we get is an antibody-dependent enhancement, because this increases the severity of the disease, okay? So let's look at the figure. In this example, we are going to be looking at the dengue virus. It's attached to the antibodies. Now this antibody is going to bind to the Fc receptors, okay? In this case, it's Fc gamma receptors. So this will be things like monocytes, macrophages, dendritic cells, so your immune system cells, okay? Now, as it attaches, it's going to get internalized, and you're going to replicate the virus. So what you get is a much higher viral load, okay? So based on this antibody-dependent enhancement, you're going to get more severe cases in the illness. So for example, you get the dengue hemorrhagic fever, where you have high fever, vascular damage. It can lead to internal bleeding and shock if it's not treated correctly, okay? And eventually it can lead to things like organ failure, okay? So it can get extremely serious if it's not treated on time. Now, how does it work? Once, like I said, after it attaches, The entry is mediated by endocytosis with clathrin-coated vesicles. Endocytosed vesicles, after the clathrin coat comes off, they fuse with the endosomes. As they fuse with the endosomes, endosome matures, you're going to get acidification, and as a result, the E dimer rearranges such that you expose that fusion peptide, okay? So this E dimer will rearrange so that your fusion peptide basically comes out, okay? It's a class 2 type fusion protein. So if you remember the lecture, see how this native conformation is for this protein? Horizontal. What do we know about the proteins on the surface of these viruses? They are not sticking up, they are more or less horizontal, okay? So again, it's going to go through this process, as you guys have already seen, and it will allow for the fusion. So let's look at, again, this figure on the side. Here's your virion. It's binding to the right kind of receptor, and it's going to undergo receptor-mediated endocytosis. Once you have your endosome acidified, you're going to reveal those fusion peptides. It's going to fuse with the endosomal membrane, and it will release the capsid into the cytoplasm, where it becomes uncoated. From there on, you can carry out translation and replication and start assembling the virus. And after that, it goes out of the cell through exocytosis. And we're going to go into the detail of this process in a minute. Question?
Yeah. How do you make vaccines for these viruses?
You take small surface take the surface protein and cleave a part of the protein that stays consistent, that's conserved. So you'll make an antibody against that.
But doesn't that cause problems?
It can. It can cause problems, okay? But there's so much of those antibodies, and usually there's other drugs involved as well. But if you have those antibodies, like it's going to bind all that, and it might prevent infection. Or it might have a minor infection as compared to a full-blown infection, okay? All right. Any other questions? Okay. All right. So if you look at the genome, once it's in the cytosol, your RNA gets bound by ribosomes and is translated. But again, it's going to produce a polyprotein which will get cleaved into precursor or active proteins, okay? Your capsid precursor, your ANC, is inserted into the ER membrane as the protein is synthesized. There is a 20 amino acid signal on the C terminals of this protein, and that gets cleaved by the ER resident protease or signal peptidase, which cleaves that signal, okay? And this basically releases the polyprotein from capsid precursor protein. So let's look at the figure. Here's your C terminal, or sorry, here's your N terminal, here's your C terminal. This is sticking into the ER lumen as the protein gets synthesized. And what you're going to cleave is here through the signal peptidase, okay? Now, the N terminal of the signal sequence on the cytoplasmic side, which is right here, is later cleaved as well. So this is done by, again, viral protease or proteinase, NS2B, NS3A, and as a result, it releases mature C protein, so mature capsid protein, okay? Now, after your capsid protein, so this is your capsid protein, this part right here, you also have your precursor M protein, membrane protein, okay? So it has a transmembrane domain at C terminus, followed by, again, a signal sequence. And this signal sequence is required to insert that E protein into the correct orientation in the membrane itself, because you want the domain sticking into the right part of the cell, okay? So as a result, what you need is these signal sequences that will orient your protein in the right way, okay? So here's a signal sequence right by the transmembrane domain. Now your PRM, the precursor M protein, associates with the E protein in the ER, and it forms a heterodimer. And this protects E from premature conformation change, okay? If you prevent that, you're going to prevent exposing of the fusion peptide as well, okay? At a later stage in the virus replication cycle, your precursor M is cleaved by a cellular protease. It's called the furin protease, and it releases this PR peptide extracellularly. So remember that this is going to be released extracellularly, not in the cell itself. It leaves the M protein associated with the virion and forms another E homodimer. Okay? So this allows for all the proteins to come together and package correctly once it's in the right environment. Now, if you look at the non-structural proteins, again, it results in formation of RNA replication complexes, so RNA replicase complexes, okay? Now, again, RNA synthesis happens in membranes in the cytoplasm. And again, you need a minus strand, so you need the antigenome of the positive sense RNA molecule, okay? So see the theme here, you have a positive strand RNA genome, you need to make an antigenome. Then you replicate that to make more positive strand genome, okay? So you get new plus strands, and they are used for 3 different purposes. One is you can also start translating them because they can be translated. So you make more of the viral proteins. You carry out replication to make RNA copies of the genome. And lastly, making of the virions by correctly packaging the whole virion, okay? So remember again, viral RNA is synthesized asymmetrically. There's usually 10 times more plus strand RNA than the minus strand RNA, okay? So remember I said earlier you have 1 to 2%, that's negative strand, rest is all positive strand, okay? Now, how does it work? Once you carry out the whole replication, which gets fairly complicated, so I'm not going to go into super detail, your virus assembly is going to take place at intracellular membranes, and as you go through, the virus will bud off from the ER and Golgi membranes, ER Golgi compartments, okay? So here's your life cycle again. You have your entry and attachment and entry, sorry. You carry out receptor-mediated endocytosis, fuse with the membrane after the pH of the endosome acidifies, and you're going to release the genome into the cytosol, where it's going to attach to these ribosomes bound to the ER. And as these ribosomes bind and start translating, you're going to make those ER resident proteins, okay? Now these proteins will get trafficked, go through, so here's your vesicle budding off, going to the Golgi, and from there on, it's going to become more and more mature. And at the Golgi, it's going to start to bud off from that membrane. So as a result, it's going to acquire its envelope from the Golgi membrane. And again, you have a pH-dependent cleavage, so here's your furin protease that's associated within that viral particle. And from there on, you're going to exocytose that protein, that whole particle out of the cell. But since these proteases are associated with the virus itself, you're going to cleave the PR protein, okay? So your PR domain gets dissociated, and the virus is now outside, and it's ready to go infect other cells, okay? So the particles are processed as they move through the Golgi, virus exits by exocytosis, your membrane anchor gets cleaved, and that happens just before the virion is released by the cellular furin, okay? It converts immature viral particle to a mature particle, and the PR peptide is only released once you go outside the cell. And again, that's a pH trigger. You go outside the cell, you're going to get to the pH of roughly about neutral. And that will help release the PR protein. And as a result, the virus is going to pack better, and you're going to get that characteristic golf ball-like appearance. Okay, any questions? No questions? Yeah. Sorry, say that again.
The release of the PR peptide is pH triggered?
Is pH triggered, yes. The release of the PR peptide is pH triggered. Okay? Because once you want to release the PR peptide once you're in the right environment. In this case, that's outside the cell. Okay? So see how these viruses, this is something we have seen multiple times. Different compartments will have slightly different pH in this case. And that will control how the viral protein folds or interacts, or whether or not it's active or not active. Okay? Question for you guys. How do you think people track pH changes in these small vesicles? How would you do that? Yeah. Sorry, say that again. They let the process go, okay. How would you stop the process? Okay, then what? You can see what the structures are, but how do you actually record the pH? Because remember, it can't be frozen if you want to measure the pH of the cell or pH of the compartment. How do you do that? Yeah. So I say that again. You look at the composition of the vesicle. How do you do that? So I say that again. Not really. Okay, look it up. How to check pH in a live cell? Look it up. I'm sure you guys use AI for a lot of other stuff. Let's try this. How do we check pH in a live cell, in different cellular compartments? Yeah? You use pH-sensitive dyes that change fluorescence as the pH changes. So you can actually do live imaging after adding these kind of dyes, okay? But one thing to know is that these dyes need to be able to get inside the cell if you want to do live cell imaging, okay? So you use these kind of fluorescence-sensitive dyes. You can also use some other mechanisms as well, but using fluorescent dyes is the most common. So pH-sensitive fluorescence dyes, okay? All right, any questions for flaviviruses? Yeah? Yeah.
What exactly is the purpose of the PR peptide in the first place? Is it the same as the PRN that prevents the premature conformation?
It basically prevents the premature conformation changes because PR peptide, once it's cleaved, it'll help the virus form correctly. So you form your dimers and your M protein, etc. Okay? All right, continuing on. Next thing we're going to look at is toga viruses. And there are several toga viruses that cause disease in animals as well as humans. And symptoms range from things like rashes, high fever, to things like joint pain and encephalitis, which is again swelling of the brain. But one thing to note here is that if a human is infected, the infection is dead end, okay? What that means is it can't easily be transmitted to other individuals. But spread through mosquitoes, so again, it's arthropod driven. That's the major public health concern, especially in tropics, okay? So you can't really transmit this disease from human to human, or these viruses from human to human, except through things like mosquitoes, Okay, so mosquito bites. So these are some of the viruses that you'll see in this, under toga viruses. Now in your textbook, the older version has this ruby virus, which is the rubella virus, but this has been reclassified. Okay, so it's not considered a toga virus anymore. So you have your Sin-based virus, forest virus, Ross River, chikungunya virus, and so on. But you can see the various vertebrate hosts, but look at this. It gets transmitted through mosquitoes, okay? So that's why control of mosquitoes becomes a major public health concern in the areas they are rampant, okay? All right, let's look at the structure like we have done for all the other viruses. Your toga comes from Latin, which means cloak or gown. It refers to the virus envelope. So this is another example of an arbovirus which gets transmitted through arthropods, okay? It's a spherical envelope particle, and it has a fringe of projection. So it does have spikes, roughly about, and the particle is about 70 nanometers in diameter. So if you look at the model as well as the EM image, this is for Sin-based virus. Look at the envelope proteins. You have your various transmembrane structures, and your viral RNA is inside the capsid, okay? And again, this has the capsid protein, you have your membrane protein, you have a trimer in this case of your E1, E2, and you develop this kind of structure for the whole virion. And inside, you have your genomic RNA, and again, your nucleocapsid and enveloped glycoproteins are arranged in an icosahedral symmetry, okay? For the envelope, you have 240 heterodimers of your proteins E1 and E2, and 240 copies of the capsid protein. What would be the T number here? Yeah. Sorry? T number would be 4, okay? So remember, T number is whatever number you have times 60 will give you the subunits. So in this case, it's T of 4, okay? All right, again, let's look at the genome. You have your linear sense, positive sense, single-stranded RNA genome, anywhere from about 10 to 12 kilobases, has a 5' cap and a poly A tail. So it resembles just like a cellular mRNA, okay? It encodes 4 non-structural proteins, so your NSPs 1 to 4, and then you have 5 structural proteins. So you have four non-structural proteins and 5 structural proteins. Now the way this works is your structural proteins are going to be translated from a subgenomic RNA, mRNA. What is that similar to that we saw? Where you have subgenomic RNA used to synthesize the proteins. Anyone? Yeah. Coronavirus, okay? Good. We have 1 capsid protein, we have 3 envelope proteins, and another 1 small hydrophobic protein, okay? For when it comes to your non-structural proteins, they're mostly for viral RNA synthesis. They are translated directly from the genomic RNA as a polyprotein, because this is a genomic RNA that resembles mRNA. You can translate it directly. And then it gets processed by host and viral proteases, just like we have seen for other viruses, okay? All right, let's look at the genome. And basically, this information is repeated, so you guys can go through that. But I want to show you this figure right here. So you have your 5' cap and a poly A tail. Non-structural proteins, structural proteins. Now here's your RNA-dependent RNA polymerase, but you also make this protein called P123, okay? And this is going to get processed into NSPs 1, 2, and 3. NSP2 is a protease. So see how it auto-catalyzes its own cleavage, okay? So you can start cleaving these proteins with this NSP2 protease. On your structural side of things, you have your various proteins that are required for translation, or sorry, required for packaging of the genome. Notice how your non-structural proteins are early proteins and your structural proteins are late proteins. And this is normally the case for all viruses. You make your non-structural proteins first so you can actually replicate the genome, and then you start making your structural proteins. Now from there on, you're going to make subgenomic RNA, and just to add on, your E-glycoprotein binds to the cellular receptors, and it undergoes clathrin-mediated endocytosis, okay? Now, your receptors for this virus, one is called the laminin receptor. It binds to this particular family of proteins, the laminin family of proteins. These proteins are part of the basal lamina of the cell. So this is attacking something very necessary for the cell. What's basal lamina used for? To attach to things like extracellular matrix, okay? Then you also have heparan sulfate, which is a sulfated polysaccharide, and again, usually it's attached to a protein. So these two act as receptors such that once the receptors get activated, you're going to carry out receptor-mediated endocytosis through clathrin-coated vesicles, okay? So here's your virus particle binding to the receptors. You have receptor-mediated endocytosis. And once you have the endosome and the virus particles in, your pH of the endosome drops, and that causes a change in E1, E2 heterodimer. So it's similar to what we saw earlier. You're going to expose the fusion peptide, and as a result, it will result in fusion of your viral membrane with the endosomal membrane. And what you do is you release the nucleocapsid core into the cytosol, where it becomes loose, and now you can start your next set of steps, okay? Now, once you're inside the cytoplasm, the RNA genome will get released, and then it gets translated, okay? It can actually, like the virus particle that's in the, or the nucleocapsid that's in the cytosol, it may interact with other cellular proteins to actually become loose and allow the genome to go into the cell. So for example, in SINBID virus, we have non-structural proteins that are translated as P123 polyprotein. You also have another polyprotein called P1234. And again, this happens 10 to 20% of the time, where the ribosome just doesn't stop at the stop codon. It just reads through, okay? So it's going to read through and make a longer protein, which then is further cleaved to produce other non-structural proteins. So let's look at the figure. Here's your positive genomic RNA. You'll start translation, and let's say you have a stop signal somewhere here. You're gonna make your P123, but about 10 to 20% of the time, the ribosome here will just read through and make your P1234, okay? Now, what are these proteins? What do you make? You have your NSP1, which is the RNA capping enzyme with a methyltransferase activity. So it adds the cap to the RNA molecules. Then you have your NSP2, which is a cysteine proteinase. It also has RNA helicase activity, okay? So I think we are starting to see that more and more. Some of these enzymes, they have multiple types of activities, okay? Then you have your NSP3. Again, we don't know what the function is, but it does get phosphorylated, so it probably does have some sort of function in the cell. You have your NSP4, which is your RNA polymerase, okay? Or eventually, all these proteins or this RNA polymerase will get degraded, but these are your major non-structural proteins. Now, the way it works is your partly cleaved non-structural proteins, they will catalyze the synthesis of your full-length antigenomic RNA. So here's your positive sense genomic RNA. You have your early translation, and you're going to make these two proteins, the protein 123, protein 1234, okay? Your P4 is basically RNA-dependent polymerase, okay? It carries out proteolytic cleavage, so you're going to cleave here, 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 and now this is forming the functional protein, okay? When it goes to transcription, again, you're going to make subgenomic RNA, but in this case, you have late transcription, which happens through a double-stranded intermediate structure, okay? You make your positive sense subgenomic RNA, you get your late translation, you make your structural proteins, and these are processed by peptidase, signal peptidase, as well as proteases in the Golgi, okay? Now, the way it works is this will start synthesizing your antigenome, but your P123 gets proteolytically processed. And that leads to switch to the positive RNA synthesis. So how does that work? Let's have a look. All right, so you have your genome. You're going to make your polyprotein. So in this case, if you go through read-through, you're going to make 1, 2, 3, 4. Otherwise, you're going to make P1, 2, 3. Now, again, you have a cleavage at the NSP4. But one thing you would notice here is that this NSP4 binds to the 3' end, as you can see here. This will allow for replication, okay? But you also have various other cleavage sites that are going to bind to various different sequences on the antigenome RNA, okay? So you cleave, so trans cleavage means cleaving by different molecules. Cis cleavage means basically cleaving itself, okay? All right, so you make this, the proteins get processed, you make your antigenome RNA. From there, you can replicate, make genomic RNA. You can carry out transcription to make subgenomic RNA, okay? So how you process the proteins will determine how you make or how you replicate the genome, okay? Or whether you replicate the genome or transcribe the genome. So in this case, you're making subgenomic RNA, which will now get translated into your various structural proteins, okay? Now, the structural proteins themselves, they are cleaved during translation, and they're directed to different cellular locations. You generate a polyprotein, which then gets post-translationally cleaved by different signal peptidases as well as furin protease. And as they go through the Golgi, you add palmitic acid to these proteins. So these proteins are, some of these, or these structural proteins are palmitoylated as they go through the Golgi. So again, looks very similar to what we saw earlier. You have your capsid protein, some domains sticking inside the lumen of the ER, and as the protein is processed, it transfers through the Golgi, where it gets cleaved, it gets glycosylated, and you add the palmitic acid moiety to that, okay? Now, this is your trans-Golgi vesicle. It's going to go to the cytosol or go to the membrane, fuse, and nucleocapsid will attach to those proteins, and the virion will bud out, okay? So here's your capsid protein that you make here, along with the RNA, is going to form the nucleocapsid. Nucleocapsid is going to go through, and remember we talked about clustering of the proteins at the plasma membrane earlier in one of the earlier lectures, that we saw how the budding happens. So you have the clustering of these proteins in the plasma membrane, which will interact with the nucleocapsid. And as it interacts, it's going to start forming the bud, Eventually, it will pinch off, and you will release the virion into the extracellular side, okay? And this is now free to go and infect other cells, okay? Everyone clear? Just one last point to add. The capsid protein domain has, there's a domain in the capsid protein that actually binds to the packaging signal on the RNA genome. And as it interacts and the nucleocapsid forms, the genome gets enclosed in that nucleocapsid, okay? So here's various proteins for SYN-based virus. Capsid protein, which forms the nucleocapsid. E2, which is the precursor to, or PE2, which is the precursor to E2 protein, which is the enveloped glycoprotein. It gets palmitoylated as well as glycosylated. E3, again, this is the amino terminal part of E2, which has the signal sequence, okay? Here's your E2, again, carboxy part of the E2, component of the projection. You have your small protein called 6K protein. This is a membrane-associated protein. It's also palmitoylated, and it has a signal sequence for enveloped glycoprotein 1, okay? The role for this protein is basically it's thought to enhance the assembly and budding as well as maintaining the pH, so transfer of protons, okay? And lastly, you have your E1, which is your type 1 transmembrane glycoprotein that's embedded into the envelope, okay? All right, so now you have made your proteins, you replicated your genome. The point now comes where you have to get out of the cell. So your capsid proteins will interact with the cytoplasmic tails of the enveloped proteins that have been embedded into the plasma membrane. And as that happens, the virus will exit the cell through budding. Now this is free to go infect other cells, okay? And again, here's the life cycle. You guys can look through the detail, but more or less similar to what we saw for other viruses. We have flavivirus, and what other virus did we discuss today? Picornaviruses, okay? So all these viruses, they work very similarly, okay? All right, any questions? All right, so since we have the exam next week, you will not have a quiz, okay? And we'll start with negative strand RNA viruses. What I'll do is I'll do a shorter lecture next week, okay? So we'll do a short lecture. I know you'll be anxious to go home and study for the midterm, but if you studied for your quizzes, it should be a review for 5 lectures, okay? Everyone clear? All right, I'll see you guys next week. Good luck. Thank you. And I will have extra office hours. I will post that for you guys later today. And one last thing, guys, before you leave, I will post your assignment questions soon as well, okay?